Hi everyone, and I hope you're well. If you're anywhere close to Cape Town, you don't need to be reminded that it is exceptionally hot today. But at least now, as the evening sets in and starting to cool down a little bit, we are experiencing a little bit of a heat wave. But luckily, like I said, it's starting to get cooler now. So, you know, clinically, we constantly see something very puzzling, right? We see patients that can share risk alleles. We even share some of the same genotypes in a family, of course, and then yet we show different outcomes. So even more striking is in the case of monozygotic twins and how they can be genetically identical and still develop a completely divergent phenotype. So these observations all point to regulation above the DNA sequence. And so today is an introduction to something called epigenetic regulation of expression. So that's, in other words, that's the mechanisms that reshape gene activity without rewriting the DNA letters. And we're lucky enough to be joined today by Professor Stephen Jeffrey. Stephen, how are you doing?
Yeah, good. Thanks, Chris. It's very hot in Cape Town, so I'm sure you're feeling the heat there yourself. But yeah,
Absolutely.
looking forward to it.
Absolutely.
Yeah,
Stephen,
yeah.
I see you're joining us you're bringing a very interesting perspective today. It's your camera is sideways for some reason.
Oh, are you? Thanks.
Here we go.
Sorry about
No
that.
problem. Brilliant.
Uh,
They always
I'm
say you must look at a problem
so
from a different lens. Say again.
I'm charging my iPad, so
Oh,
the
I
charger
see.
is to the bottom of the iPad.
No
Um,
problem.
so let's
No
do
problem.
balance. It's okay. No, we're good. We're good. There
No,
we go.
excellent.
That'll work. That'll work. There we go. Good. All right.
Excellent. So let's jump right into it. So, what is epigenetics, right? So, epigenetics is the study of mechanisms that cause chromosome associated and heritable changes in the gene expression. So, but importantly, and the most important thing is that it's not dependent on the changes in the DNA sequence, right? So, it's changes to DNA expression without changes to the underlying sequence. And the epigenome, because you'll hear a lot about omics and the omics revolution. That's genomics and epigenomics and transcriptomics and all the omics. So the epigenome is the pattern of those modifications in a cell at any given moment. So over a lifetime, a cell has only one genome, but it can exist in many epigenomic states that are changing as it differentiates and adapts and responds to environmental cues. And why clinicians should care about this is because epigenetics explains how cell types become different without changing the DNA. So it's tied, which is tied to disease mechanisms, especially cancer. And it reframes how the environment and behavior can shape biology. So, crucially, unlike DNA mutations, or epigenetics and epigenetic changes are potentially and in bold, potentially reversible, which is why they're actually a major therapeutic target or therapeutic focus. So let's actually delve a little bit into the details there. So there are three major epigenetic mechanisms. Um, oh yeah, we'll cover all three of them. So DNA methylation is the first one. Histone or chromatin changes is the other one, and non-coding RNA regulation. You might as well actually just say RNA regulation. It doesn't specifically have to be non-coding RNA, but RNA regulation. So let's start with DNA methylation. So like we just said, we have all of these omics, so genomics, transcriptomics, epigenomics. So there's another one which is called the methylome. Methylome. So that's, as you might have guessed, all of the methylated nucleotides present in a genome at any given time, the methylome. So in mammals, methylation occurs after replication and during differentiation. Just a little bit of chemistry. So methylation is adding a methyl group to a nucleotide. What is a methyl group? It's a carbon. It's essentially, well, depending on where the carbon is placed, if you let's take one of the nucleotides specifically. So let's take cytosine. If you add a carbon group to that, you have 5-methyl cytosine. Bit of chemistry. So methylation occurs almost exclusively at something called a CpG dinucleotide, right? Often clustered together in something called CpG islands. And these are, if you think about a DNA sequence, this is in a region called the promoter region, right? They're also found in regions called upstream regions. So, a simple clinical rule of thumb from this framework is that promoter or promoters and CpG islands are usually adjacent, so next to essential and cell specific genes, because the cell specific genes themselves tend to be unmethylated when activated. So, and you can think why, because methylation can and often does silence activity. So, when active methylation in a promoter or a CpG region links or partially blocks the transcription factor binding, and so can affect the expression levels, usually lowers it. Doesn't have to specifically, but as a rule of thumb, usually lowers it. But yeah, methylation does occur beyond promoters. So you get, uh, that's an important nuance. So you get most methylated CpGs aren't next to genes. Many actually sit in these repetitive heterochromatin, including regions like centromeres. So this is where methylation contributes to silencing as well, and it helps to prevent transposons activity and genome instability. So the distinction between those is that in the one case, you have a case where it's physically silencing the gene, and the other one is a case where it's actually mechanically constricting histone. So that's histone modification. But both of them are actually examples of methylation. So methylation patterns are established and maintained by enzymes called methyl transferases. Um, and so demethylation, whether it's passive or active, enables epigenetic reprogramming of gene activity, either globally or specific to a single cell, right?
Chris, I'm so sorry. I've got some technical issues. Yeah, I'm back now. So, um,
No problem. So we quickly spoke about a couple of different forms of epigenetics. So we spoke about, or there are three different types of epigenetic changes that are common, right? So the first one is DNA methylation and
yes.
demethylation. Then there's histone modification, and then there's non-coding RNA. So we've spoken a little bit about methylation now, and in a nutshell, it's essentially when a one of two things: either when a carbon binds to a nucleotide and causes that gene to be silenced or expression to be dysregulated, or there is a histone modification version of it where it binds to the chromatin region in cells, and that can also physically change, mechanically change expression levels and transcription factors. So the other, that's methylation. So the other type of epigenetic regulation is histone modification or chromatin remodeling. So DNA regulation can be physical. Literally, DNA wrapped around nucleosomes is not equally accessible. So you get these remodeling complexes which can shift nucleosomes. So we have quite a few omics tonight. So we've had genomics and epigenomics, we've had methylomics. So these are essentially whenever there's the suffix omics behind anything or ome behind anything, that's referring to all of, say, genomics, for instance, is all of the genes in a cell or all of the genes in an organism. Epigenomics is all of the epigenomic shifts in an organism. And I think it's important, Steve, a point that you may have not been here for is that a person is stuck with their genome for their entire life, but the epigenome can change consistently throughout one's life. So, um, yeah, so we were talking a little bit about remodeling complexes and how these can shift nucleosomes and histone tail modifications. So essentially, what happens is methylation can bind to these histone tail modifications and either loosen or tighten the chromatin. And when what that does is it turns the regions either more open or closed, and transcription literally becomes easier or harder. Whether it's usually correlated to the looser the chromatin is, the easier transcription is to happen um, in that area. And this is due to protein or different types of enzymes and proteins being able to access all the different regions more easily. So histone markers or marks are written and read and erased by all of these little dedicated proteins that flow into and out of the cells. And these marks can be reversible. And that reversibility is where the clinical context becomes extremely important because that leaves us targets for potential therapy, right? So the histone code, right? So, okay, that's a little bit of theory. So the histone code is this idea that combinations of histone modification within and between different histone proteins actually control the transcriptional status. So that's that what I tried to use the analogy where it loosens and tightens. So the shorthand encodes that histone, or the shorthand encodes histone name, amino acid position, and modification. So if you were to write out what type of histone modification occurred here, you would say something like H5K27 ME3. So that would indicate where is the methylation and what amino acid position changed and how does this methylation or modification look. So amino acid and position, um, and of course histone name. So that's the nomenclature that you use to indicate, okay, this is the histone and how it's been changed. So
So if we have to take it down to absolute basics, we'd say like the like the hardware and the sort of epigenetics concepts are all the software that feeds into the how it all works. Would that be a good way of working, or was that a little bit too simplistic, Chris?
So because of I'm experiencing a little bit of an internet delay here, so I only caught the latter half of it. So are you asking whether the epigenetics is almost like the regulatory side of the hardware, which is the genome?
Yeah, yeah,
Right. Yeah,
yeah, yeah.
I would say
That's
so.
right.
Yeah. That I think that's a great analogy. I think that's a great way to describe it because if you think of would you start with a genome or genotype versus when you end with a phenotype, which is the ultimate
Hmm.
expression of all of this. Between this and that point, there's actually a lot of transcription, environmental factors, metabolomics. There's so much in between that happens, methylation, that even though the blueprint, the genome or the genotype is saying that this is what should happen, what you should see, what you ultimately end up seeing is not necessarily that. That's why we get cases where identical twins who have monozygotic identical twins, of course, have exactly the same genotype, have very different expression in terms of, or can have very different expression in terms of their behavior and how they come out and how they look and how what they do. Not so much how they look usually, because these things also, of course, accumulate throughout your life. But so whether you're comparing young people or old people is also going to be a big difference. But I think a good analogy or a good example to use is twins, one of which had a healthy lifestyle and one of which had a not so healthy lifestyle. And you can see the effects that this has on things like aging, for instance. And it's a good
Hmm.
indication that, your genome or your genotype are the cards that you're dealt, but the epigenome and the environmental and behavioral impacts play a massive role in how that ultimately dictates how you move forward from there. So,
Yeah.
and whenever people to ask me about genetics, I love using the analogy of poker, right? So if I can be dealt a 6 and a 7 in my hand, so it's a terrible hand, or could be a terrible hand. But if there's a
Hmm.
8 9 um what's a Jack Queen King on the board, then I have a straight,
Yeah.
right? So
Yeah.
beautiful. So yeah, so it's not just the hand that you're dealt, which is your genome, it's also the environment that you happen to find yourself in. Right. So, but
Yeah, it's a lovely analogy because absolutely, because you might find yourself finding external circumstances suddenly working well towards your, you know, towards winning the whole hand. So
Yeah,
I like that.
absolutely.
Yeah,
Absolutely. And of course,
yeah,
pocket aces, the other way around. You can start with pocket aces, but then around you sit people with full houses and royal flushes. So so you get both sides of
yeah,
that
yeah.
coin.
No, no, no.
Yeah.
I see you're quite a keen poker player yourself, so it's I like that.
No,
So,
no, no, not. You know, it's been a while since I've played.
yeah,
Actually,
yeah. I
terrible.
got you. I got you.
We should
But,
play sometime and then gamble a lot of money because I'm terrible. The
so
right, so
all right, no,
yeah,
but
yeah.
it's a fantastic analogy. But I mean, and I think I mean, do you think it's becoming more accessible to really manipulate this? You know, because I mean, we can do a lot to our own genes. I mean, it ties into multiple layers of not only medicine but health. You know, I mean, you've got the extreme side of it, you've got the cancer side of things, but then coming through to modifications in terms of just someone wanting to modify their risk of Alzheimer's, you know, with a
Mm.
very generic supplement of, of some description. So it's a very exciting part of life, really. I don't want to say medicine because, but yeah, I mean, where do you see the most exciting evolution at the moment in epigenetics?
Yeah, so it's because it's a so I think one of the breakthrough moments is the fact that we realize that epigenetics has a mechanical underlying biology to it. So for the longest time, there were geneticists that almost treated epigenetics a little bit like a pseudoscience. Even now, there's some of the old school people that don't actually treat it like a validated science that it is, which I think they're going to have a very hard argument to win there, considering that I think it was either last year or the year before the Nobel Prize was given to the people who discovered the function of microRNA, right? Which is another type of
Okay.
epigenetic regulation, which is coincidentally what was very much what my own master's revolved around as well. Um, but
So is that microRNA? Sorry to interrupt, but was microRNA a relatively new sort of area of epigenetics? Was it not known
yes,
until quite
yeah.
recently?
Yeah, or the functions of it. So the methylation was one of the first ones. So just adding carbon groups that in promoter regions was one of the first ones that we kind of figured out, and then we could also see the mechanical histone modification. But um, non-coding RNAs actually regulate gene expression as well. So this would be long non-coding RNAs or microRNAs or something called siRNAs, where I think it stands for short inhibiting RNA or something like that. Uh, and essentially, what these do is through different mechanisms, they can either trap transcription factors or adapters that scaffold these complexes. And usually what they do, I'll use the example of microRNA, is they bind to something that's supposed to deliver a final product and degrade it or silence it or changes how it affects. So I think microRNA is actually the easiest one to describe. So it's if you think from the genome level, when you have the DNA, then the messenger RNA goes from the DNA towards the ribosome, but the microRNA binds to the messenger RNA, and it brings in an enzyme called RISC, so RNA-induced silencing complex, and this usually cuts the messenger RNA, and so then because the blueprint is cut, the protein doesn't get expressed as much. So this is one, and so obviously you
Oh,
can see
okay.
from that. Yeah, yeah. So obviously you can see from that then
No, that's
the
exciting.
sequence there. Yeah.
So
Yeah, it
that's
is
with
exciting.
that one. Would microRNA potentially be easier to manipulate because it's still a bit further downstream, or do you think?
Yeah, absolutely. So that's where we are. So we've there's a lot of tests, and I know this is a little bit of a long-winded way to answer your question on the
Mm, mm.
therapies of it, but it's so we're at the point where we're testing injectable microRNAs and actually using these as an actual treatment. Um, and this is quite exciting because we're also in the realm of developments in genetic editing and genetic engineering being of such that you can take engineered microRNAs and maybe if you see, because you use the example of it, was it Huntington's or use one of the neurodegenerative disorders? I think it was Huntington's that used it as an example.
Mm, mm,
So we're at the point now where you can potentially take this microRNA, which is usually approximately 12 nucleotides long, and engineer a specific sequence that will target um, a specific set of messenger RNAs and be able to regulate expression. Um, but keep in mind, so microRNAs usually cut or degrade um, messenger RNA. So the mechanism there that you're essentially tapping into is something that'll decrease expression. So if you're sitting with a disease that's caused by increased expression of certain genes, that's great because then you can target it this way. But if you're sitting with diseases that are linked to some increases and some decreases, that becomes a bit more difficult. So the interesting thing about microRNA and non-coding RNAs is that these actually tend to affect multiple spots on the transcriptome. So one single, in other words, and simply put, one single microRNA variant can bind to dozens of different messenger RNAs linked to dozens of different genes, right? But that actually also complicates using this therapeutically because that means if you administer microRNA that decreases the expression of one gene, you're also decreasing the expression of other genes. So
mm, mm.
it's complex. It's possible, and in
Yeah.
theory, not more than just possible. In theory, and in lab practice, we've done this. We've administered all sorts of non-coding RNAs, and we've seen we've seen expression changes because of them. But to
Mm,
actually get to the point where we have a clinical treatment based on that, we're getting there. But it's we that's where we are now. We are getting there, but we're not
mm.
there yet.
Wow.
Yeah.
It's an
So
exciting part of life. I mean, and it makes a lot of sense to target disease with it, cancer, genetic disease. But what about the really big diseases? I read a book a little while ago, but about longevity, and they were talking about the four horsemen, the four big diseases that really are causing people to die. I mean, we've
mhm.
really done a lot in terms of longevity with infectious diseases.
Yeah,
But if you look at things like cancer, obviously we've spoken about cancer, but heart disease, diabetes, Alzheimer's, those are the things that really do take most people out eventually. Do you think,
yeah.
and where do you think, I mean, in terms of cardiac disease, where do you think we can start modifying cardiac disease with epigenetics? So are we anywhere close to that? I know it's maybe a little bit of a slightly new concept, but do you think we could cardiac heart disease or diabetes, or even Alzheimer's? What are your thoughts on that, Chris?
Yeah, no, so the metabolic syndrome related diseases like diabetes and all of the cardiac ones, absolutely that would be a major one. And from a theory perspective, there's no reason to believe that there wouldn't be um, many different treatments for these things. And similarly for the neuropsychiatric disorders, there's lots of studies on the way of trying to we're still in the phase of mostly identifying in the case of microRNA, is involved in all of these systemic and complex pathways. We're still identifying which methylomes, so gene sets of methylation, are involved with increased or decreased expression, and how this correlates to disease expression and because as you know, of course, as a researcher, a lot of these you're either talking about the treatment or you're talking about the onset of the disease. So we're still in the phase phase with those things of really trying to figure out why is the disease presenting the way it is. So what is linked to the disease presenting as we see it before we can even get to the point of treating it. Um, and with a lot of these things, so the reason, yeah, so we are tend to be a little bit further along with treatment and therapy when it comes to cancer. That cancer research does tend to be a little bit of a pioneer with these things. And so cancer is increasingly understood as both a disease of genetic and epigenetic changes, with epigenomic alterations emerging as a major pathway forward in the sort of initiation process. Um, but it's no, I believe, and this is what I think. I believe that neuropsychiatric genetics and cardiac and all of these is short to follow, right? So we'll start seeing more and more treatments in these spheres as well. But what we see practically and clinically is still mostly in the cancer and oncology spheres. Um, but it won't be long. I'm certain of it. It won't be long before we see a lot of developments in cardiology and neurology, and any diabetic related fields as well. The four horsemen, as you said. Um, yeah,
Yeah,
but
I
right
mean,
now, yeah,
yeah,
yeah, yeah, yeah, go
yeah.
for it.
I mean, so we're absolutely, the old scientists and the old doctors in Pharaoh's days, and that they were always looking for the elixir of life. But I mean, we're much closer to that now, and if you can really get rid of those prospects I mean, longevity will. I mean, it'll be who knows where it'll end. So that's very
Yeah.
interesting thought.
Yeah, so there was all sorts of research on anti-aging and the links to epigenetics and it's also the studies are also still in their infancy, and whenever someone speaks about whenever someone promises to expand or extend lifetime, of course you have to put on skeptical goggles and that type of thing. But I read a very interesting book by someone called David Sinclair called Lifespan. And so they actually did a lot of research they're there in Australia, but I think they have a lab in Harvard as well. But they they did a lot of research into the epigenome and how this correlates to lifespan and how basic behavioral things like exercise and diet and these things affect your epigenomic profile, and how ultimately you can slow down aging is essentially what the book spoke about. And
Yeah.
they're a
No,
big proponent for a supplement called NMN, which is nucleotide. No, it would be nicotine mononucleotide, um, nicotinamide mononucleotide, and essentially where this would fit in would just fit in with the carbon acid cycle. So if you think about the sort of the Krebs cycle, the creation of ATP, it would essentially help with that cycle from a molecular point of view. But yeah, they're a big proponent for that and how that is correlated.
sounds like
Yeah, I'll go for
yeah.
it.
So
Sounds
and
like.
isn't that vitamin B? Isn't nicotinamide? Isn't it one of the vitamin B complexes? Or I mean, so do you think people have been onto something yet taking vitamins? I mean, it's I've
Isn't that niacinamide? I might
oh,
be incorrect.
okay, yeah, no, good, thanks for correcting me there. Yeah,
I'm
um,
not sure. I'm not sure.
yeah.
Yeah.
So um, so I suppose that, and I mean, I found the whole concept of longevity very interesting. And there's a lot of people who are taking all sorts of supplements, but it sounds like just in some of my more recent readings, things like creatine, the use of omegas. I mean, omegas they sail to the top every time. Omega 3.
Mhm,
People
mhm,
always, and that's been for at least 10, 20 years. People have known about omega 3s,
mhm.
and then a little bit of magnesium. They say is quite good for you.
Yeah.
Do you have any? Have you, in your reading, found anything that you are very convinced about? Because there is a lot of stuff you can take, and I've never found anything that finds that vitamin supplements really does much to prolong your life,
Mm.
or even do much for improving your quality of life. I don't know. What are your thoughts
Yeah.
on a very interesting topic?
I agree completely, and I can't say that I found anything that I'm very convinced about. I I take actually take NMN every day, and every now and then I would take a little bit of omega 3 and magnesium because these things are healthy, but they're not often the way they're sold. It sounds like if you don't take these, you'll inevitably go everything will go wrong that could possibly go wrong. So these things are healthy, but they're not. they're not as healthy in the sense of they're not these miracle supplements that a lot of people say they are. If anything comes close to a miracle supplement, it's creatine because of the also the effects that it has on cellular energy, ATP. But that's all it really does. And then similarly to you, I'm not very convinced that vitamins in pill form that you ingest really do much um, or are really as useful, whether it comes to their correlation to health or a lot of people, say take your vitamins so you don't get sick, but even that correlation is questionable. It's like the oldest example of this is people swear by vitamin C for a cold or infectious disease, but the correlation, statistical correlation between vitamin C and immune system is it's not that strong. So I don't know why
Yeah.
people latched
Well,
onto this. Yeah.
I don't know the evidence, but yeah, people are. I mean, it was just something so strong. I mean, since we were growing up, your mom would give you a glass of orange juice if you were sick, or
Yeah.
you grab some oranges. And it was very much a winter thing where I grew up. But
Yeah.
I have actually been reading a little bit more about vitamin C recently, and in fact, the place where it has got some really good evidence is in skin renewal, skin
Oh
regeneration.
yeah. No, that's a different story.
So
That
yeah,
makes sense.
so
Yeah.
that's where I think it is really been shown to be useful, and I mean that's also another completely different conversation, and that's
Yeah.
where there's a lot of potential marketing commercial value. If one could really
Yeah.
look at skin regeneration and making people look younger,
But
because that's a huge market. But
would that not be topical or would that be ingested? Um,
topical,
topical.
topical
Yeah.
vitamin C cream, and then
Yeah.
retinol, which is vitamin A, I
Of
think it
course.
is.
Yeah.
Yeah,
Yeah.
yeah. So,
Yeah.
so those are the two that, in my reading, sail to the top in
Yeah.
in terms of daily
Absolutely.
applications. And
Absolutely. Although that's not,
yeah,
I wouldn't say that's epigenetic, or maybe it could have a link that I'm not aware of, but that's mostly cosmetic in terms of, I know retinol, for instance, causes a little bit of a little bit of a inflammation effect going on there, really. And it does
okay. So
clump
it's not.
out.
It doesn't. Okay. So it doesn't really exert its influence through genetic components. More like a regenerative cell. But doesn't regeneration of the cells all feed back into the genetics?
Well, it may. If it does, then it's not something that I'm aware of. So yeah, I can't comment on
Okay.
the molecular pathways. I wouldn't be surprised if it also does that, but I don't know.
Yeah.
So yeah, yeah, yeah. But
To
yeah,
to take
yeah,
this
yeah,
to a yeah. Carry on.
yeah.
Yeah.
No, no,
No,
yeah.
no.
You
Please
want to
carry on.
take it? Yeah. No, I was just gonna say. So
No. Well, I mean,
yeah,
no. Please carry on. Sorry.
yeah. So I was gonna take it back to cancer actually, because a lot of the treatments for these things actually do come back to what the field of cancer pioneers. So if you're talking about epigenetics and hypomethylation, um, well, you get so you can either get hypomethylation, so that's unmethylated or undermethylated areas in the gene sequence, and then you get hypermethylation, which is over methylated areas. So cancer often shows something called global hypomethylation, right? So which can activate. So you get genes that either silence genes, or you can genes that upregulate other genes. So promoters versus silencers. And specifically in cancer, where this becomes extremely relevant, and we spoke about this 2 weeks ago, I think, is you get tumor suppressor genes, which are the type of genes that actually help, um, you know, usually through the process of apoptosis, it regulates that cancer or rapid cell proliferation doesn't happen. But so when you silence these genes via hypermethylation, you can actually be suppressing a tumor suppressing gene. So you're suppressing the gene that's supposed to be suppressing the cancer related things, and then you can get cases where the mechanism built in to stop these rapid proliferation of cells doesn't activate. So where this comes in is that with targeted cancer therapy or epigenetic therapy, these specific epigenetic sort of markers are actually reprogrammable. So you can take methyl transferases, so that's an enzyme that reduces methylation, and then you can actually affect. In fact, not only does it reduce methylation, it also affects acetylation. So you can acetylation is pretty much the same thing, except it's not a methyl group. It's a different chemical group that's bond to the DNA, and it causes the chromatin to sort of be silenced because it constricts it literally. But deacetylation makes it express more. It desilences it. That's just two examples of how an epigenetic therapy can be used to affect biologically cancer, your natural sort of cancer treatment a little bit better, or your natural systems to prevent cancers a little bit better. So
Oh,
yeah, it's a good and yeah, and the age old case with these type of things is whenever we talk about preventative medicine, it's very difficult to get people to fund that or companies to fund that, and it's very difficult. People don't often come to prevent diseases. And this is once again, as we always come back to genetic screening comes into play. So when you know you're at high risk, then these sort of prevention methods might become very applicable in the future. But we just don't have the broad sort of screening for everyone yet to even implement the second step, which is then, okay, also let's implement long-term epigenetic therapies globally to try and prevent all of these different, in this case, cancers and diseases that are correlated with it. But it is a lot less invasive than something like we know there's famous examples of someone being at risk for breast cancer and then doing a mastectomy just because of the high
yeah,
risk.
very common.
Yeah,
Yeah, no,
yeah,
that's big. That's a really big thing. And people are finding themselves to have BRCA genes and straight into but double mastectomies in young women, and if they can really get rid of that risk, I mean, that might be one of the first places to focus to try and modify that gene expression because those of women are well. But and I suppose how to triage or strategize in terms of which to focus on. I'm sure scientists and those guys like you working on this ultimately have to see where it does make economic sense. And the big cancers, breast, colon, um, melanoma, um, cervix, and ultimately, yeah, I mean, one. And I think it would be great to see some advances in that world because it's we see a lot of women getting that, um, and it's often a little bit too advanced and it's quite um, defacing on the patients as well when they have their surgery. Um, so that's quite a good one.
yeah.
It'd be nice to see some progress in things like pancreatic cancer as well. That's a really devastating diagnosis too. So it'd be nice to see if there would be some movement somewhere on that level too. So
Mm.
I'm glad to hear that the movement is very much in the cancer world, but and I suppose that's what's slowing a lot of things down is that there just isn't enough money going because the pipeline is still very long.
Yeah,
It's not
that's
like
exactly it.
they'll do research now and find an application for it in the next 2 or 3 years. It sounds like we're still ways off, I think.
Yeah, for some of
Um,
it. So there's
do you know of
there,
any?
yeah. Do I know of any?
No, no. I think I want to hear what you're saying about keeping on the cancer train.
Yeah, yeah. So that is the thing, exactly as you said. So we have a very focused approach now, and I think that's actually why people focus so much on cancer, because a lot of times what we find in cancer research can be applicable to other diseases and disease profiles as well. And so yeah, so you mentioned pancreatic cancer. So we do have these large epigenomic mapping efforts going on to sort of try and generate reference epigenomes across all sorts of different cell types. So whether it's in the pancreas or the lung or wherever you might be looking at different cancers. So And this combines methylation to histone marks to chromatome accessibility, transcriptomes, non-coding RNA. Those are the microRNAs and those siRNAs and things that we spoke about. And then to clinically see mechanistically how or understand them. How are these contributing to complex diseases like we mentioned Alzheimer's and all of these cancers, so pancreatic cancer, and then to employ precise targeting of the regulatory layers that drive these diseases. But yeah, it is ultimately going to be a focused effort first, and so we do see most of the movement happening in monoallelic expression. So that's essentially when you where only one allele, so only one of the genes, so to say, that you inherited from a parent is affected or active. And so it's a lot becomes a lot simpler when you only have to do or when you only have to target one allele. So whenever you target a gene where both sides of the chromosome is affected, it becomes more complex. And whenever you deal with something that there's multiple genes or multiple SNPs or multiple variants or all of these multiple epigenetic markers and promoters and transcription factors and transposons and different cell types and environmental types and behaviors and all of these things that come into play, it's an exceptionally complex field. And then some tissues are also just inherently more complex than others. So neural tissue is difficult to even model. We've tried to make neuro organoids in petri dishes, and we've tried to get appropriate models for this type of thing, but it becomes an exceptionally difficult thing to study ethically as well. And so that's another reason why cancer treatments is the focus, because we have the foundation, we have the model, we have the cell lines that we can use for these things in abundance, actually. So, which is much we can of course tissue engineer and grow any tissue basically, but the pipelines to do studies are also just so much better defined in something like cancer that it almost becomes the default sort of we want to study epigenetics broadly, we want to study methylation or histone code or whatever. Let's look at this cancer cell line first because it's what we have, and then hopefully we can apply it to something down the line later on. And we do end up usually applying it to more and more things from there. But we are, like you said, very much still in the phase of let's do this first, let's focus on this first. And then inevitably it will be applied to all of these different places as well. But right now we're still figuring out the how it works. So we haven't even completely figured out how it works yet. And when we do that, then we can start figuring out, well, what do we target and what do we treat from there? You know? So yeah, so one of the most clinically resonant themes is that uh, early life environment, like say maternal nurturing, can correlate with epigenetic differences in promoter region methylation and histone acetylation patterns. So my own masters looked at this as well. It was quite interesting how stress or especially high stress like childhood trauma can greatly increase epigenetic modulation and epigenetic expression of genes. Um, so anytime you have these sort of early life interventions with any sort of epigenetic cause, uh, what you see is that in later life and onset, or yeah, in terms of disease onset later in life, you can actually have uh, a much larger disease risk and risk profile. Now that goes as with complexity of these genomics and epigenomics and all of these things. It sometimes goes both ways. I looked at some markers that we didn't report on because the correlation in our small sample set wasn't statistically significant enough. Um, but we did find indications that sometimes you get resistance factors from something like childhood trauma towards some, but it's none of this is published on because It's well, this is published on just not in our studies, but the yeah, because you get sometimes you get that methylation, so that would decrease expression relates to increased disease risk, or you can get that methylation, which leads to decreased expression, leads to decreased disease expression. So it's a very complex, very complex thing to deal with because whenever you look at epigenetics, you're inherently not only looking at the genome and the underlying genetics, but you also, in a way, quantifying environmental factors. Um, and it becomes a phenomenally difficult thing to treat and look at. Uh, come to think of it, yeah, 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 yeah. You
Yeah,
wanted to say
and I mean,
something?
look in my field, having been an obstetrician as well, there are many things that we struggle to explain, but start to make a lot of sense if you look at it from an epigenetic point of view. You know,
Mm,
so you'd have risks to all sorts of things in childhood, or birth, around birth, the delivery, etc. And simple things like prematurity and diabetes or hypertension. And then it also comes into the discussion around the role of delivery versus C-section versus normal birth, and we know that when you have a normal delivery, the baby gets exposed to the mother's microbiome in a
mm,
much more extensive way than a C-section, which is a much more sterile delivery. But that microbiome has a very powerful epigenetic effect on the baby in its very early phases of life. And as you can imagine, the huge exposure that a newborn fetus comes under in those first few days and weeks of life, I mean, suddenly they're in a world where there's all these powerful environmental influences on them. And so we
yeah.
are now starting to try and understand exactly, what we have done by promoting C-section versus normal delivery. And, I think we'll maybe be able to explain that a bit better as we get the epigenetic influences on these patients.
Yes, I absolutely agree. And along with that, not just epigenetics, but all the things we're learning about different microbiomes and bacterial influences on these type of things. We're also because as the more we learn about epigenetics and genomics of people, we also know that on your skin and in your gut and in all of these places you are outnumbered by all sorts of different bacteria by millions to one. um, and so we do research. It's called metagenomics, or meta epigenomics if you want to look at the epigenomes of things like I don't think there's anyone looking at the epigenomes of multiple thousands of different bacteria, but it becomes such a complex thing because as you mentioned now with the C-section versus say a natural birth, you don't necessarily know which environmental effects are causing all of these late onset factors. And that I do think I agree with you. I do think that's definitely something that could become something that we sort of epigenetically have do in a study and or maybe even screen for and see, okay, well, this is the case that you've been exposed or not been exposed to this type of thing. Can we treat it? And if not, can we at least learn from it for future generations to know that what should we do instead? Um, but yeah, it definitely becomes a a big thing. I wonder, so do you often hear, I would almost say, stories of say, let's take the example that you use. Let's take a C-section versus not. Maybe even anecdotally, do you see a difference in how the effects of it, or not really?
No, not firsthand, but there's a lot of discussion around this, and a very topical discussion in modern obstetrics is how you can colonize the baby with the mom's bacteria again,
Yeah.
because it's very well documented now and understood that, and there's data on that, looking at the outcomes of fetuses or babies that are born by C-section versus normal delivery, and it's overwhelmingly um, indicative that a baby born by normal birth does better. Just from a overall point of view, just take the same genetic potential in each baby, but
Yeah.
although a baby born normally would have a better chance at some things.
Yeah.
Um,
Oh, okay.
yeah.
Is that speculation or is that definitely the case?
No,
Is
no,
that
there's evidence. There's evidence for it, Chris,
oh, wow.
and they're taking it down the line um, of the you know the influence of the microbiome,
Yeah.
and that is there is some influence there.
Yeah.
Um,
Yeah. Absolutely.
in keeping just before we get to the end, in keeping with all of this, obviously a lot of people asking questions around the role of epigenetics in those dread diseases that you diagnose prenatally.
Yeah.
Uh,
So
and
that
I
would
think,
be
yeah. So,
so
I
like
mean,
Beckwith Wiedemann syndrome and Prader-Willi and
oh,
Angelman and those ones. Yeah.
yeah, I mean, there's quite a large array of these things. And, if they are diagnosed antenatally, some people would see it as a reason for termination.
Yeah.
And I think it would make a huge difference to families if there's a way of altering the outcome of those babies in utero. There is a lot of work happening in utero, and we've got some exciting work that will be happening soon in Cape Town for fetal
Oh yeah.
surgery. But
Okay.
what are your thoughts on the modification prospects of in an antenatal situation or prenatal situation, the baby?
Yeah. So that is a very complex case. So if you, especially if it's a case where you can early on affect if you can affect the transcription or expression now before it's caused harm by epigenetically switching on or off
Mm,
the level of
mm,
regulation, that would be amazing. So
mm,
in theory, we can do that. Um, but if it's usually if it's developmental, or in the case of say something like X chromosome inactivation, to create something called a
mm,
bar body, it becomes
mm,
very difficult to to do anything about it. You can't really treat it when an entire chromosome, for instance, has been silenced. Um, so
mm,
yeah, so it may in the future it would be great if we can not only screen for but also do something about it. But it becomes very difficult because a lot of these cases, even if we screen for it, a lot of the quite severe disorders are you can't really treat for them with basic sort of methyltransferase treatments or that type of thing because it's an entire chromosome. So yeah, we're not really at that point
yeah,
yet where like
got you,
I would
got you.
love to hear at some
Yeah,
stage about this surgery, but we can't, unfortunately, we're not genetically able to treat those diseases after they've been screened, and they are present. It's just
yeah.
not
So
viable. But
yeah.
yeah, so
So trisomy 21, I mean, that would be very difficult to sort out antenatally. I
yeah,
mean, because that's
yeah,
obviously one of the ones that you are the most fearful of. And when we're getting older now, the risk is high. That's not
yeah.
something that epigenetics will be able to fix soon.
No,
That you can imagine.
not soon. In theory, you could you can cause a synthetic bar body by inducing a lot of long non-coding RNAs and then deactivating that additional chromosome in theory, but I don't know of any clinical practice. And I'm sure there's all sorts of different reasons why people aren't doing that or haven't done that. Um, yeah, so I'm not aware of any, but yeah, it'll be extremely difficult. We're not there yet. We can't remove or deactivate or add chromosomes just yet.
Yeah.
um,
I'm just
yeah,
putting it out there, just raising the the potential and the need for this kind of therapy, and hopefully one day we'll see movement in that
yeah,
direction as this
yeah,
science of epigenetics starts to get more used for good, rather than people trying to prevent hair loss or
yeah.
trying to a little bit younger or trying to get taller or whatever.
Absolutely, yeah,
Hopefully we can channel it right. You know what I mean?
yeah. No,
So
absolutely,
yeah,
absolutely. And we it's always so interesting because we do find all of these sort of things that even I look at it, I'm like, this is so cosmetic. Why are we? But I, at the same time, then from that, similar to the cancer research, we do find a lot of things that ultimately, oh, molecularly, this does make the cell younger, uh, at least in the way that we measure it.
Yeah,
So you do find these sort of interesting outcomes from it, at least. Um, we should ask
yeah.
your good friend, Professor Peter de Jong or Dr. Peter de Jong sometime because he's obviously more on the obstetric side. So we should ask him about the effects that we see and whether the difference between something like a C-section or not has led firsthand because he would be the one that does see the sort of firsthand effects of these things and whether he
Well,
thinks
yeah.
yeah
Well, I
hello
mean, I'd like to refer people to our webinars on the Pelvic Foundation and Professor
yeah
Phil Steer, who was the previous editor of the British Journal of Obstetrics and Gynecology, gives a great talk on this actually.
is it
And
oh
being from the UK, yeah. So being from the UK, where they do push C-section, well, they do promote vaginal delivery still very much. There's some strong reasons for that as well. So yeah,
okay. Yeah, that's very interesting. I hope we
yeah,
hear more about that on the Pelvic Floor Foundation of South Africa's webinar. Um, yeah, that would be
yeah,
amazing.
yeah. We'll try and get him back to talk about that.
Yeah,
You've really raised a very important subject. Anyway,
yeah,
Chris, thanks so
awesome.
much.
Thank you very
Was
much.
there anything else you
Yeah.
wanted to bring up?
No, that was my little Chris talk for the night.
Yeah, I mean, this was a primer. I mean, it's without a doubt the most exciting part of medicine. I mean, it's we can look at everything, but it makes a lot of the other advancements look quite small. Robotics or whatever else we're dealing with. But I mean, this is huge, and medicine will look different in 50 years' time. It's going to look different, and this is the reason it'll look different, and
Yeah,
that's what excites me. So,
yeah,
but
it
thanks so
excites
much.
me too. Thank you very much. And yeah, thank you for listening tonight. Right, cheerio.